heads up, he's promised he's going to cooperate, but I've got a 5-month-old at home. So if you hear some screaming, that's what's going on. So yeah, I'd love to talk to you today about some of the work that I've done during my PhD and postdoc, sort of spanning a broad range of topics, but in general relating to how we can think about engineering applications to epidemiology, which is an area where I think there's a lot of room for exciting research and where a lot of good work can be done. So just to sort of contextualize what we generally think of as challenges to global health, I think there's been this picture that's been emerging of sort of, because of improvements in sanitation and technological development, top leading causes of death in high-income countries in modern day times are typically thought of being related to non-communicable conditions such as stroke or heart disease. But it's important to recognize that there's still a substantial burden of death in low-income countries that's coming from communicable diseases. But as you're all probably aware, this can be turned on its head pretty quickly. So this is data from a couple of weeks ago where the leading cause of death in the US was a new respiratory infection that you've probably all heard of called COVID-19. That's very much a communicable disease. So I think in bioengineering, there are tons of exciting technologies that we've developed for both communicable and non-communicable diseases that we learn about, and examples that come to mind would be design of biomaterials, diagnostic devices, other diagnostics and other devices. At sort of the more cellular level, we can think about with studying within host dynamics and more aspects sort of at smaller length scales, and of course vaccines and therapeutics. But one thing, at least to me, that was never made as clear as I've sort of learned more recently from the postdoctoral work that I've been doing is the impact that bioengineering technologies can have on population level transmission when we think about communicable diseases in particular. And I think there's actually a fairly big gap in terms of mechanistically how this is achieved, and one that I think would be really interesting to spend more time thinking about as biological engineers. So in particular, when we think about population level transmission, one very classic framework that's used in epidemiology is known as the Susceptible-Infected-Recovered framework, or SIR, where we think of the population as being split into 3 bins, essentially those susceptible to an infection, those that are currently infected, and those that have recovered or are immune. And sort of the main ways that I see as bioengineering technologies being able to affect this transmission chain would be either through affecting the transmission rate directly, which is the force of infection from infected individuals to susceptible ones, or by essentially changing the composition of these bins. So moving individuals, say, from the susceptible to recovered class without necessarily needing to go through the infection, which is what, say, something like a vaccine would do. So it's impossible to give a talk on anything related to epidemiology these days and not mention COVID-19. So I thought I would sort of ground this framework in some of the open questions these days that we're hearing about in order to contextualize it, and then apply it to the research that I've done afterwards. So thinking about issues to susceptibility for COVID-19, one question that comes up repeatedly is what fraction of the population is still susceptible. And then by the other side of that coin, what population is immune, what fraction of the population is immune. And then other questions that arise in the context of immunity. So I was just going to say that in order to determine this, likely technologies like serological testing, measurements of antibodies are going to be critical in order to answer these questions. Thinking about immunity, though, what does immunity mean for COVID-19? And that's a big question that's going to need to be addressed sort of from a bio and epi perspective. For instance, are you immune once you've had the disease? Can you be reinfected? How long does your immunity last? And that's all sort of active areas of research at the present time. Related to how we can think about the transmission rate of being affected, we could ask questions like, what's the effect of different policy measures on transmission? So we've heard a lot about social distancing. That's why we're all doing this remotely today. There's been a lot of active work characterizing what effects of social distancing might be on this force of infection and how that might be expected to change future infection burdens, or policies such as mask wearing, which I've been thinking about more recently. And finally, of course, thinking about transmission, ultimately, the development of a vaccine would be critical, and also in terms of moving people to the immune or recovered class. Okay, so I think now that we sort of understand the framework that I'm sort of thinking about these questions in terms of, I'd like to contextualize the work that I've done and show you how it all in general relates to these sort of major underlying themes or questions. So my graduate work focused on one aspect of susceptibility, which is related to the innate immune system, and one important characteristic in particular, or one important component being mucus. So I spent a lot of time doing rheological characterization of this material, and subsequently using that analysis to understand the potential of mucus to be used as a diagnostic biomarker. And I did so with cervical mucus in the context of preterm birth, which I'll talk to you about today. I also did a bunch of work with saliva and its potential as a diagnostic, but I'm not going to have time today to get into the details of that. But I put the references there, and I'd be happy to talk more about it later on today. Also related to susceptibility, more recently I've been thinking about more classic within host dynamics models and how cellular susceptibility may play a role on disease progression, as well as population level susceptibility, in particular to the introduction of a new viral strain into a population. In terms of transmission, I've done work in my postdoc thinking about how climate may modulate the transmission of infectious diseases, both in terms of vector-borne diseases and directly transmitted diseases. And finally, I've done some work on thinking about aspects more related to public policy and human behavior, and that in particular, how that might affect vaccine adoption in terms of affecting the immune class of individuals. Again, I'm not going to focus on that today, but I think it's an interesting area of research where there is room for engineers to take part as well. And finally, I'll wrap up by talking about future areas of research that I'm excited to continue and would be excited to continue at McGill. In particular, thinking about new bioengineering technologies as can be applied to tuning transmission rate of infections, and in general, information management and policy advising for disease. So diving into the first aspect then, which is mucus and the innate immune system. So you're going to hear probably more about mucus today than you thought you were going to, just a disclaimer, but it's a very important biogel in the body. It coats every wet, non-keratinized epithelial surface, and all of these surfaces are sort of shown on this man or human figure here. The main component of mucus is a large glycoprotein called mucin, and the body produces about 1 liter of mucus every day. So that's a pretty substantial amount. And what's interesting for this material is that its biochemical structure has an important role in dictating its mechanical properties that in turn allow it to serve different functions in the body. And to that effect, it really does have different properties and serve different functions depending on where it's located, and the body manipulates that locally. So for instance, in the eyes, we have the tear film, which is a very watery mucus layer that serves to hydrate and lubricate the ocular surface. And then in the stomach, there's a thick static mucus layer that protects your epithelial cells essentially from ingestion by harsh acidic juices, gastric juices. So this biogel can be tuned depending on the environment, but pathogens and diseases have figured out that this can be done as well. So sort of one of the prototype diseases when we talk about mucus would be cystic fibrosis, and here because of dysregulation of sodium channels and possibly mucin overproduction, respiratory mucus becomes very sticky and difficult to clear through normal mucociliary transport, which can lead to biofilm formation and ultimately deadly bacterial infections. Another example would be a stomach ulcer-causing bacterium called H. pylori, which can actually change the viscosity of the gastric mucus layer and colonize underlying cells. So in order to think a little bit more about how this happens, let's talk about this major building block in a little more detail. So as I mentioned, it's a very large glycoprotein, about 2 megadaltons in molecular weight for just one subunit. It's got an amino acid backbone, and then this characteristic feature of these little red things are sugar chains that stick off radially like a bottle brush. So in solution, these aggregate into even larger macromonomers through sort of quasi-covalent bonds, and then form a physically associated network that's maintained by electrostatic repulsion between the charged sugar chains and hydrophobic interactions between the non-glycosylated portions of the molecule. So what I did a bunch during my PhD was to use a model system for native mucus that was slightly easier to work with experimentally. So what we did was to purify these mucins from animal mucus tissues. Here I'm showing you MUC5AC, which is a mucin purified from stomach mucus, but it's also found in other parts of the body. And then we reconstituted gels from this underlying building block that we could tune depending on what we added to the solution. So for instance, by changing the pH, we could modulate the charge on the polymer backbone, which changes its conformation. By adding surfactant, we can target hydrophobic interactions in the gel. And by adding salt, we can change the Debye length by screening electric charge. And so we can we can modulate the degree of electrostatic repulsion in that sense. So what basically our technique was to tune the environment with small external changes and then see how that affects the composition and the mechanical properties of the gel. So the main change that I'm going to talk to you about today is what happens in response to a pH modulation. I won't go into the others in detail, but I'll put up a summary at the end, and then I'd, of course, be happy to talk about anything in detail later on. So macroscopically, what we observed and others had observed before us as you decrease the pH of the gels is that they become stiffer. So this is shown here. This is a classic rheometric plot, log-log. And what you see is that as measured by the elastic modulus and loss modulus, these increase as the pH is lowered. And so a classic sort of polymer physics picture would then suggest that maybe what's happening is that as we decrease the pH, the mesh size or the points on the mesh, the distance between connections is decreasing and the gel is becoming stiffer by some biochemical mechanism. And so that gives us a first axis that we can think about in terms of what's happening to the rheology of this system is that macroscopically, so what we observe at the large scale comparable to the length of our measuring device, which is on the order of about 10 millimeters, is that decreasing the pH somehow is increasing cross-linking interactions and causing the gel to become stiffer. But as I mentioned, that's a macroscopic picture of things at the length scale of a rheometric device, which is quite large. And in particular, if in bioengineering, since we tend to be interested in very small things, it might be necessary to revisit this picture since the mesh size of mucin that we're talking about is on the order of nanometers to microns, which is comparable to the size of a lot of small things that would be of great interest to us, such as viruses, drug delivery vehicles, bacteria, a whole host of other particles. And so in that sense, if the probe that you're using to measure the properties starts to become comparable in size to the mesh size of the gel, the environment that that probe is seeing might actually be quite different than what you observe at a large scale. So what I mean by that is that below critical size, rheological properties start to depend on the length scale at which you probe them. So in order to assess this, we used what's known as single particle tracking or multiple particle tracking since there's many probes. We inserted micron-sized tracer particles into these same gels and we measured their mean square displacement over time. And what you're seeing in this plot is an ensemble average over many particles. And interestingly, what we observed, which was sort of in stark contrast to the macroscopic picture, was that the pH2 gels, which were ostensibly the stiffest, the particles were able to diffuse to the greatest extent in them, which is sort of counterintuitive. So a first clue as to what was going on was to say, let's not look at the ensemble average, but now let's look at every single particle track. And what you see is this quite stark increase in heterogeneity as we drop the pH, where at pH 2, some particles are freely diffusing. So this would be a slope 1 for these fastest moving particles. It's just slightly slower than they would move in water, whereas other particles are barely moving at all. They seem very confined. So visually, we wanted to assess this, what was going on as well. So we added small positively charged fluorescent peptides, which bind to the many negative charges on the large mucin molecules, and they are fluorescent. So under the microscope, we could see where they were aggregating in order to get a better picture of what the gel structure was. So in order to I'll walk you through this experiment just in order to show you what we were looking for. So we tried this in pH 7 in buffer. So this is just peptides in buffer, and you can see they're very homogeneously dispersed. Now, if we add mucin to the gel at the same weight percent we were considering before, again, not much exciting has happened. They still seem very homogeneously dispersed. pH 2 buffer, same picture, but now when we add mucin at pH 2, you see a very different scenario where it looks as if we have regions of very dense fluorescence, which would suggest dense mucin regions where these peptides are binding, and then there are other regions that are essentially void of mucin. So coming back to this picture that I showed you before, while macroscopically, we saw that decreasing the pH had effect on increasing macrorheological stiffness. Microrheologically, it had the effect of increasing heterogeneity through a phase separation that was induced from, again, change in the polymer conformation and probably the inducing of additional hydrophobic cross-links. And so playing with these axes then of adding surfactant, adding salt, we were able to sort of piece together a more holistic picture of how the gel forms and how it can be manipulated by the environment. So that concludes my rheological characterization of mucus portion. I'll talk a little bit about the diagnostic work that we've done now, and that was in the context of preterm birth, which is defined as birth prior to 37 weeks of gestation. It affects over 12% of pregnancies in the USA. A lot of these stats will be USA specific since that's where I've been for the last 7 years, and is a leading cause of newborn mortality. And there are some sort of estimate measures for predictive tools for preterm birth, such as cervical length, but they tend not to be robust. So there is a clinical need for predictive diagnostics and biomarkers. So one hypothesis as to what could be occurring in this condition is that the cervical mucus plug, which separates the sterile uterine environment from the colonized vaginal environment, is supposed to serve as a protective layer, essentially, to keep pathogens out, with the thought being that if there is colonization by bacteria or other pathogens, this could trigger the early delivery. And so one hypothesis was that in a high-risk pregnancy, this barrier may become more permeable, allowing the pathogens to go through. And there was some work that was done before I started working on this project that had shown that macroscopically there is some evidence of changes in mechanical properties between samples from low and high risk pregnant women, both in terms of shear rheology as well as extensional rheology, which is when you stretch a material between plates as opposed to deforming it in shear. So we decided to look at things on the smaller scale. As I discussed before, again, adding these 1 micron chaser particles to the gels. And what we observed by looking at the mean square displacement is that, first of all, between ovulating non-pregnant women and pregnant women, there were statistically significant differences in particle mobility, and that the particles were much more confined in samples from pregnant women, which makes sense. During ovulation, this barrier is supposed to be the most permissive in order to allow for fertilization. But at this scale of 1 micron, we weren't able to see statistically significant differences between low and high risk samples, although there was a trend of decreased mobility in low risk samples. So that's still fairly large, 1 micron, when we're thinking about the mesh size of native mucus. And we can think about a number of ways in which a barrier could be exclusive. Steric hindrance is what a 1 micron size would think about more. Are you physically too large to fit through the mesh that you're trying to pass? But there are other mechanisms as well. So for instance, a particle could be smaller than the mesh, but it could have some surface biochemical properties that would cause interaction with the components of the gel, and that could also prevent it from passing through. And so to that effect, we decided to use these same reporter peptides as I mentioned before, and this is work that I did with my previous colleagues from MIT, Kate and Jacob. So using both positively charged and negatively charged 10 nanometer probes, so now we're a couple orders of magnitude smaller. What we saw was that cervical mucus from low risk patients was significantly better at excluding these charges, these peptide probes, than samples from high-risk patients. And so it suggests that potentially there is an important role for biochemical changes to this barrier in the context of preterm birth and high-risk birth. Okay, so that's going to wrap up my whirlwind tour of mucus, and I'm going to switch gears to talking about another aspect of susceptibility now, and that's more cellular level susceptibility in the context of within host models for diseases. So the disease that we've focused on for this work has been measles, which is a leading vaccine preventable disease. So even though most of us have been vaccinated, I say most of us since there's been a resurgence of anti-vaccination sentiment, but regardless, there's still about 100,000 deaths every year from measles, mostly in children under 5 years of age. So from an epidemiological perspective, vaccination has had a big impact on population dynamics. And here I'm plotting data from the UK. Vaccination was introduced in the late 60s, and you can see that this affects the number of cases, but also the dynamical timing of cases in terms of seasonality and sort of yearly occurrence. And again, related to what I was saying before, this from the dynamical model, this comes from moving individuals from the susceptible to the recovered class without having to pass through the infected stage. Even though this vaccine is very effective, it's interesting that it's not exactly known from a biological perspective why it achieves the high level of protection that it does. The other paradoxical thing about measles is that the wild type infection is very robustly immunizing. So that's why it serves as the prototypical SIR model, since there are other diseases where you might actually be susceptible in the future, in which case we might need to add additional bins to these models, since you might not necessarily remain in the recovered class for life. But measles is the prototypical disease that behaves in this way, and this drives the interesting dynamics that this disease exhibits at the population scale. But despite this, it targets lymphocytes in the body. That's the way that the virus manifests. And as a result, people who are infected with measles lose immunological memory that they've acquired to other infections. And so sequelae, you can develop secondary infections or sequelae even to things that you've already been infected with. And this poses an additional public health challenge. So this is shown through two different recent studies here, which suggest that the burden of measles is actually larger than just the case number of measles every year. So this is a lot of aspects to think about. One area of this problem that we've been focusing on recently is how baseline immune activity might affect clinical measles severity. So what I mean by that is that I mentioned that measles virus targets lymphocytes in the body. In particular, it targets activated memory lymphocytes, which express the cell receptor CD150 or SLAM1. And in general, when your immune system is activated, this is when you would have heightened expression of this receptor or heightened lymphocyte activation. And so the question that we posed is, well, how does the reverse apply? If you are, say, fighting a previous infection and your immune system is in a more activated state, can that go on to affect the severity of the progression of measles? So this was done in terms of experimental data in collaboration with some folks in the Netherlands, Rick and Rory, who use animal models to think about disease progression. So they have data on, first of all, infection in different animal models, but also they had some interesting anecdotal evidence that where they inserted things like temperature probes or other sites of vaccination of other viruses into the animals, so where there was likely inflammation already, there would be a lot of presence of virus in those areas, which suggests active infection. And that's seen here as the fluorescent areas, since the viruses that they use are typically have EGFP in them. The other set of data that we had to work with was an interesting serological data set from a cohort of children in a Dutch Orthodox community in the Netherlands who were infected with measles. And so the researchers had blood samples pre and post infection that we were able to analyze in order to think about how their immunological memory to other infections may have been affected as a result of the infection, specifically by looking at their IgG antibody repertoire. So some IgG antibodies are disease specific in the sense that once you've been infected with something and your body seroconverts and you have some baseline protection to it, you have antibodies that float around that remember that prior infection and can react more quickly if it comes back. So what we were doing is we were looking at how that repertoire of immunological memory changed pre and post infection. So I think this is sort of an interesting question for a number of other diseases as well. Just a quick side note, coming back to COVID-19, since it's on my mind a lot these days, I think that it's also an interesting question to ask in this case. So in particular, the renal angiotensin system has been identified as potentially playing a role in the progression of COVID-19 through the fact that ACE2 receptor acts as a binding site for the spike protein of SARS-CoV-2. And so there's a lot of questions now about how baseline ACE2 expression might affect the severity of COVID-19, since we know that some individuals tend to experience much more severe infections than others. So this is an active area of research now, with people thinking about potential age and gender differences in ACE2 expression or genetic variation between populations, and also potentially predisposition due to things such as smoking or comorbidities such as COPD. So back to measles, what we did to think about this was to develop a within-host model for the immune system. This is a very sort of baseline approach, which we're hoping to expand on subsequently, but we're thinking about the immune system as a well-mixed pool where we have susceptible lymphocytes that can become infected by the presence of virus, then it can go on to produce more virus. We also have activated T cells that can kill infected cells. And we also have plasma B lymphocytes in this mix, which are the ones that secrete the antibodies that are already in place to prior infections. And this is what we use in our model in order to assess or to predict what changes in IgG repertoire should look like pre and post infection. So we find with the model that as potentially, perhaps unsurprisingly, on the x-axis here, as you increase the number of available susceptible cells, or essentially the fuel that the virus has, the peak infection conditions are worse as measured through max viral titers, max number of infected cells, and also the loss in IgG antibodies at the start of infection compared to 60 days post-infection. And there's some evidence of this from the experiment as well, and experiments as well. So in animal models, there is evidence. This is data on T and B cells from different lymph nodes in macaques that were infected with measles. And what you see is that when you have increased cells of memory phenotype, which are the ones that express CD150 and can be targeted by measles, in general, this is associated with increased levels of cellular infection. And there's also some evidence of this in terms of the human cohort, although it's still a small number and needs some additional work to think about this, but there is some evidence as well of increased antibody loss during more severe infection, and that this may be associated with an increased baseline cellular susceptibility. So although this is still in the hypothetical sort of hypothesis stage, we do think it has some interesting basis, especially in terms of thinking about why the take of measles vaccine might differ between individuals. So some individuals seroconvert very well and seem to achieve better levels of immunity than others. So this begs the question of whether sort of underlying immune system status may play a role in that. So the next epidemiological aspect that I'll talk to you about incorporates two of these bins. So I'm going to talk to you about dengue virus, both in terms of population level susceptibility and in terms of potential effect of climate on transmission. So this is connecting aspects from susceptibility and transmission rate. And obviously, I think there is a lot of overlap when we think about questions in this framework. So in terms of how climate can affect transmission, there are mechanisms for both vector-transmitted diseases and directly transmitted diseases. So vector-borne disease transmission means that there's an intermediate between humans that are infected. So a lot of diseases may, for instance, may be spread by mosquitoes. Ticks would be other potential vectors. And so temperature and other climate parameters play a role in regulating the length of some of these parameters, such as the extrinsic incubation period, or the time between when a host takes a blood meal from an infected human to when it could transmit to the next infected human. different aspects of both the parasite and vector life cycle are sensitive to temperature, and this has been shown in a number of studies. And also precipitation, particularly for mosquito vectors, plays an important role since their reproduction is dependent on the availability of water. In terms of direct transmission, there's currently a lot of work trying to understand the burden of whether aerosols or droplets play a bigger role in respiratory transmission, but in either case, the surrounding environmental conditions such as humidity levels and temperature play a role in how quickly droplets might evaporate into aerosols, and that can affect transmission dynamics. And then also virus survival on different surfaces has been shown to be sensitive to climate as well, or the environment that is occurring in. So the mechanism that we use to think about this is to take this continuous time SIR model that I showed you before and to discretize it into a stochastic time series framework instead, where we can come up with distinct values for the transmission rate at different time steps. And this tends to be a good approach when we're thinking about epidemiological data, which doesn't occur continuously in time, but typically you get data every week or every bi-week or every month. So this is a framework that helps us take something from the continuous frame to a more discrete time frame. And then what we do is we develop different regression models to relate the transmission rate that we observe back to different climatological data. So as I mentioned, the disease that we thought about this in the context of was dengue, which is the most clinically important vector-borne disease in terms of burden on the population. And from a virological perspective, this virus is interesting because it exists in 4 distinct strains. And infection with one strain infers lifelong immunity to that strain, but not to the other 3. And actually, second dengue infection with a different strain can be worse because the body is primed from the first strain, and your response to the second one can actually be very serious. And typically, a lot of worse outcomes like hemorrhagic fever or shock syndrome would be related to secondary infections. And the data set that we had to analyze was a large dengue outbreak that occurred in Sri Lanka in 2017. And so we wanted to use this data set to tease apart different potential roles for climatological, virological, or sociological drivers on the epidemic. So from a climatological perspective, actually Sri Lanka was a very interesting place to do this analysis because it's a very small country with a large amount of variation in climate. So here what I'm plotting is an entropy metric, which when this value is small, it means that there's very minimal seasonal variation over the year. But when it's large, it means that there's very noted seasonality. So towards the southwest, you can see that precipitation levels are more constant throughout the year, whereas as you move towards the north and the east, there's very much more distinct seasons of very wet and very dry periods. And just even at this quite crude superficial level, we could see that precipitation over the year was leading dengue cases by about 2 months, and that there seemed to be distinct timing between precipitation and case burden, whereas temperature is quite warm and not too variable over the year. And so once again, turning to these regression models, what we showed was that climate can play an important role in establishing the timing of annual dengue transmission in Sri Lanka, but the climate of 2017 wasn't sufficiently anomalous to justify why we would have seen such a big epidemic that year. So this allowed us to turn to other potential drivers to think about why this may have occurred. In particular, hospital reports showed that dengue 2 may have reemerged after a period of being absent for about 7 or 8 years, which tends to be quite a typical characteristic for multi-strain diseases such as dengue. And so what we did then with this model was to think about how climate and strain replacement can act synergistically, in particular in the future, to think about how this might be predicted to affect disease burden. And so by plugging in climatological values for the future using large-scale climate change models that our climate science collaborators have developed, we were able to assess how potential changes in population level susceptibility in combination with changes to the transmission rate driven by climate could be expected to change the timing and numbers of disease in the future. And we found that they could act synergistically to yield a greater number of cases than you might expect from a single driver alone. In terms of directly transmitted diseases, our group has also thought about respiratory syncytial virus. So this is a leading cause of lower respiratory tract infections in infants and responsible for about 5% of under 5 deaths globally. And what we found was that, sort of similar to the dengue story, epidemics exhibit distinct seasonal cycles. And this is actually very reminiscent of what happens with influenza, where in temperate locations, there's peaks in winter months, whereas in more humid locations, there tend to be epidemic peaks during the rainy season. And so again, developing these climatological models where we input precipitation and specific humidity, we're able to make predictions for the burden of how RSV might change in the future. In this case, we did it in the context of the USA and Mexico. And now what we're doing, and sort of a lot of other people are trying to do as well, is apply these models to think about how climate might affect COVID-19 case burden. And sort of the question that keeps coming up is, will the summer months save us, and will everything disappear by then? And there's a lot of variation in what predictive models might say across groups, but I think undeniably the fact that such a large fraction of the population is susceptible still will have a huge impact on driving dynamics, potentially even more than changes in transmission through climate. So that's the whirlwind tour of the different aspects of the work that I've done. And with the last few minutes, I wanted to talk a little bit about future research directions that I'd be interested in pursuing at McGill. So I spoke to you already about how climate can modulate transmission rates. And one aspect that I've been working on these days is how human behavior and public policy might be able to do so. So as I mentioned, how does social distancing and mask wearing, how can we expect that to curb transmission? But I think there's a lot of room for interesting bioengineering technologies that could play a similar role too. So thinking still about respiratory tract infections, one interesting aspect of this puzzle that's emerging is that infection that's associated with the upper respiratory tract tends to favor transmissibility. So it seems to be easier if I cough or sneeze for you to catch the infection if it's colonized my upper respiratory tract, whereas infections that seem to target the lower respiratory tract are more associated with pathogenesis or have greater case fatality rates. And both of these impact what's known as R0, which is something you probably heard about a lot on the news recently, which is the number of secondary infections that arise from an infected individual in a totally susceptible population. So we're just starting in the epi literature to see some models that connect this within host picture to the population level dynamics by thinking about where the virus is infecting, and then how is that getting into emissions from the respiratory tract, and then how is that going on to affect R0. But I think there's a lot more room to be done in this area, specifically in terms of mechanistic or fluid mechanical models to think about what exactly is being emitted, what is the viral concentration, how does this depend on immunological aspects of the specific virus in question. And I think I would be very interested in looking at things in this respect. So again, in the context of COVID, just because I've been thinking about that a lot recently, I think, as I mentioned before, there are some clues as to what might be happening in terms of the infection and disease transmission framework. So thinking a little bit more about what cell receptors are playing a big role. Are there aspects of the respiratory system physiology that could make some infections worse than others? For instance, is mucociliary clearance hindered in particular individuals, or is lung physiology changing? Is that playing a role? And so I think all of this again relates back to the work that I had done earlier on mucus and the fluid mechanical aspects of that in terms of respiratory physiology. And I think there's a lot of interesting stuff to do there. And then also, again, because I've spent a lot of time thinking about mucins, but I'm also open to other biopolymers, I think there could be room for designing materials that can bind and interact with viruses and potentially mitigate or reduce infection to underlying cell layers. And in that sense, alter the within host kinetics and the viral infection dynamics in an important way. And lastly, motivated by some of the projects I've been working on more recently, I'd like to continue the direction of sort of data science and policy advising that I've been working on. So one aspect of that has been through the climate and disease group at Princeton, where we've been using a bunch of data analytics techniques in terms of, in collaboration with climate scientists, hydrologists, a number of experts from a number of different fields in order to think about disease transmission in that way. And recently I've become a working group member of the DELF, which was put together by the Royal Society to think about how to use data analytics to tackle different aspects of COVID-19. So I'd be very interested in pursuing that in collaboration with a whole bunch of different groups at McGill and my existing collaborations, and also continue to think about that in the sphere of policy advising as well. So with that, I'd like to thank my groups from MIT and Princeton and all the funding sources, and I'd be very happy to take any questions from you.
Okay, clapping hands now. Okay, so we just start with the question from Amin. So can you see it, Caroline, can you see it in the chat?
1 second.
So you click on chat.
Right. Oh yeah, here, yeah. How does my work on mucus rheology contribute to improving mucosal delivery of vaccines? Yeah, so that's an interesting question. I haven't thought about, well, at the time when I was doing my mucus work, I hadn't thought about vaccines much in particular. One thing that we did think about a lot was drug delivery vehicles. And I guess if we could find a way to encapsulate some aspect in a small particle that we would deliver through a mucous membrane, that would be something I've thought more about. But again, as I mentioned towards the end, thinking about how mucins and other sticky polymers might bind to viruses and other small things could be interesting. But in the sense of drug delivery vehicles, that's actually a really big challenge and a huge focus of work right now, because mucus is so sticky and good at excluding things. So some approaches include surrounding the vehicle with small polymers that stick off that can sort of get through the barrier more easily as opposed to binding through biochemical interactions. There's been a host of techniques that have been developed. Even the particles that we were working with for our particle tracking posed a number of challenges. So we tried pegylating them and changing their surface chemistry. So that would sort of be the extent of what I've done, but I'd definitely be more interested in thinking about that in the future.
Okay, Caroline, you see that there are other two questions for Max and Alan.
Yes. So Max says, when designing these complicated multivariable transmission models, how can you resolve the effect one variable may have on another, such as climate on policy? Yeah, so that's definitely a challenge. And I think that's coming from an engineering background. Maybe that's one thing I didn't expect about epidemiology, is how interconnected everything is, and the difficulty of, causality is always something that's difficult to show. So I think the approach that we took in the climate models was just to develop very strongly qualified regression models where we had a whole host of dummy variables, and we could control for things that were changing yearly or even throughout the year that were probably not related to climate. And we do so even at the expense of probably taking some of the power out of our system by probably some of the effect of climate is being lost in this mix in order to control for these different factors. But yeah, I think there's definitely going to be a lot of overlap, and that's a challenge that we're encountering these days in, say, the assessment of masks or other policy. Just because masks are implemented on day one and there's a change after day one, it doesn't mean that it's necessarily attributable to masks. So I think that, yeah, that's something that has to be assessed carefully, for sure.
Alan, apparently you want to ask the questions.
Sure, it's just easier than typing. So first, thanks for your talk. Very interesting in a broad range of topics. So I'm interested in the hydrogel rheology and microbiology. And I wanted to ask you, one of the things you just mentioned was that you've been doing some PEG coatings as one way to, I guess, render the particles inert. Do you see any difference in your microbiology when you peg coat these versus just putting in I think it was carboxylated particles?
Yeah, so that's a great question. And I didn't peg coat them personally, but we were working with some folks at Johns Hopkins who do a lot on drug delivery vehicles. So we were getting the particles from them. One thing we found was that at this length scale at 1 micron, which is really pretty big when we're thinking about the mucin mesh, peg coating doesn't play a big role. And it seems to be that up to about 500 nanometers, you're sort of saved because most of the interactions are coming from steric and not from sort of chemical or electrostatic interactions. But definitely there's been a lot of work showing that at smaller scales, it would be very important just because mucins are so sticky, they're good at binding a lot of things.
That's perfect. So that actually leads into my next question about what you think the mesh size scale is and how that's related to your 1 micron particle size. I guess 1 micron's big,
Yep.
But if you're modulating the pH and you're expecting the mesh size to change, are you spanning that 1 micron range, or what range do you think the mesh size occupies?
Yeah, so I'm showing you just the work that we did at 1 micron, and then we have a follow-up paper that's written but not published yet, showing what happens with a whole host of sizes, so changing by at least an order, more than an order of magnitude. So I think at pH 2, the reason why 1 micron probes were really interesting is because we did see this mesoscopic phase separation, and so a lot of the changes that we were seeing weren't exactly mesh sizes related to crosslinks between the polymers, but also a result of this actual phase separation that was occurring that we visualized with the peptides. But you're right. I think if we were, and the other tricky thing is that, I've been asked a million times, like, how many cross-links are there on one mucin molecule? And we just don't know. It's such a mess of a molecule that it's very hard to say something like that. So yeah, I think your question is really well taken. And this is something that we debated Michael Rubenstein on this paper a lot about, how effective are 1 micron particles at visualizing this. I think it was helpful for the purpose we were serving, especially since we wanted to look for agreement with macro rheology. But there's a lot of questions that smaller particles were better suited to answer. And actually what we saw as we kept dropping the size of the particles in the pH 2 gels was that we started getting better and better agreement with the macrorheology, as eventually these particles were starting to get into like the actual densest part of the gel.
Fantastic. One more question, if you'll permit me. When you were looking at the change in pH and the change in rheology, so I'm interested in gel salt transitions as well. And it was a little bit too fast for me to notice, but do you see any variation in the loss tangent there as well? Do you see these are just becoming uniformly more resistive in loss and storage, or are they becoming more solid and more fluid-like?
Yeah, that's a great question. So what we were seeing over and over again was just these power law responses, which maybe you see in biomaterials as well, which seems to be just the presence of these large number of weak physical gels, weak physical interactions. We did see something like a bit of a sol gel transition as we went from pH 7 to pH 2, where it was a bit more lossy at pH 7 and less at pH 2. But it's, again, in terms of seeing like the more classic polymer physics or rheological transitions, it was a bit harder to make out. But I think, yeah, like I said, between pH 7 and pH 2, I think we could observe some of those transitions, with pH 4 being sort of a nice intermediary that we could look at too.
Great, thank you very much.
Yeah.
Caroline, Amin has another question there in the chat.
Oh boy. So I wouldn't call myself an expert at this, but there are challenges. So I think, if I'm not mistaken, the vaccine was dengue vaccine tried to be rolled out a few years ago. I can't remember exactly where, whether it was the Philippines or somewhere else. And there were a lot of clinically serious infections that resulted afterwards, because you were sort of seeing some amount of antibody enhancement because of the vaccine affecting secondary cases, or even first cases in that sense. The vaccine was almost behaving as a first infection to make second cases worse. So yeah, I think there are still problems, and that's one of the challenges with dengue, is it affects so many people, but there isn't a good vaccine at the moment. So I would say that figuring that out will be a challenge.
Although I'm further commenting on the question, actually, although there is approval with WHO and there is recommendation, and I just wanted to understand from, because, you know, it's multivalent, it's quadrivalent, there is the 4 serotypes that are represented there. Within the context of what you studied, actually, the combination of the 4, what would be the dominant problem actually to be solved to have this vaccine less controversial actually than it is currently?
Yeah, I mean, I guess ideally it's a vaccine that can target all 4 strains and not allow for reinfection by another strain, which would take out this problem of antibody-enhanced predisposition and aspects like that. And I'm not by any stretch an expert on this vaccine, but from what I understand, it didn't achieve that in some contexts. So I think, yeah, our work, you know, I didn't do a full 4-strain model, and whether 3rd and 4th dengue infections are a big deal, I think, are clinically important, is something that remains to, it maybe isn't 100% established, but in terms of at least getting rid of this second infection, whether the first one is from a vaccine or from an actual wild type strain, I think is the big challenge.
Again, there is a question from Robert.
It's time series. Yeah.
Okay, wow. So Janisha has a number of questions. What's on the chat?
Okay, what was the process of developing a model for epidemiological dynamics? So the regression-based models, I would say we only did in the context of climate, because in that sense, what we're trying to do is we're trying to relate the transmission rate at time points back to climatological data without necessarily, you know, this is a bit of the question with machine learning, is it just a black box and do you actually know what's happening? So I would say in that sense, maybe regression models fall into that category to a certain extent, but short of knowing the exact functional form of how every aspect of a disease transmission is affected by a certain climate variable, that's sort of the best we can do at the moment. So that's where those models played a role. But in terms of epi modeling and dynamics, I would say a much more classic approach is an SIR or an SEIR, where E is exposed but not yet infected, or another type of dynamical model, or some of these models that have like 20 different compartments because you were infected the first time, then you become susceptible again, but your susceptibility is slightly different relative to your baseline susceptibility. So I would say something like that. Even like it's super simple to write out, right? It's three differential equations, but the complexity of the math that arises is pretty astounding because of the nonlinearity of the system. So I think you can achieve a lot of interesting dynamics from even simple models. Next, she asked, has the effect of social distancing been explored? Yeah, so the paper that I cited, if you're interested, was just published in Science a few days ago by a group at Harvard. Kistler is the lead author. So that's one example. So essentially what these models are doing is they're saying, can we interpret social distancing as effectively decreasing the transmission rate, which you can, because if you don't meet as many people, then you're not going to transmit the infection as readily. So essentially what that can be interpreted as doing is decreasing this beta value, or this, what you probably hear of more as R naught in the media these days. And so then that changes your growth curves, that changes your force of transmission. And so, yeah, there's definitely been models thinking about how that might affect case burden in the future. And then last question was, do I see any parallels between COVID-19 and dengue? Yeah, that's a tricky question. I think you probably hear about viral evolution in the media a lot, but I think that's a different picture than having sort of these 4 distinct, somewhat stable strains that have been existing for a long time and sort of co-circulating within the population. So but I think there's still a lot to be learned about what immunity to COVID looks like. We don't know yet if you're immune after you've had it, which is why I don't recommend rushing out of the house if you have had it. There's some trials that are happening in animal models where they're trying to reinfect them, and they're looking at, even in humans, how your IgG antibody response takes place after you're infected, but there's still a lot of questions. And I don't really think that it's exactly analogous to dengue, but in the sense that viruses are always evolving, I guess there's some similarity in the thought process there.
Okay, so if we do not have any other question, I guess that it is, and in the interest of time, we should move to the teaching part.
Okay. So I'm going to just stop.
But by the way, whoever has questions, don't be shy, put them in the chat, and I'm sure Caroline would answer them in due time. Okay.
Yeah. Okay.
I just put the clapping.
Oh, thanks.
So you can do that too.
Okay, I'm going to pull up my teaching presentation now. Okay, so we're going to switch gears a little bit, and I'm going to talk to you about pipe flow of Newtonian and non-Newtonian fluids with applications to blood flow. Should I just get right into it, Dan, or?
No, go ahead.
Okay, all right. So this is motivated by the human circulatory system and what we can think about as engineers in this context. So this is pretty fascinating when we think about some of the statistics. So if you add up the total length of all arteries and capillaries and veins in the body, it reaches about 100,000 kilometers. You have about 5.6 liters of blood in the body, and a single cell in the blood is estimated to travel about 19,000 kilometers per day. So that means there's a whole lot of infrastructure to maintain, and there's a whole lot of fluid flow. And that there are obviously some issues that arise that as engineers we've done a lot of work trying to tackle. One big one would be atherosclerosis, which is related to fatty deposits starting to line the blood vessels, and that can change the cross-sectional area as well as the mechanical properties of the vessels and cause all sorts of changes to blood flow. So one big development there has been through the design of stents, which can be inserted into vessels to reopen them. Engineers have also thought a lot about the design of heart valves, whether they range from synthetic or somewhat animal-based. And this is their design is super important, because if you alter the fluid mechanics or the velocity profile of the blood flow, that can have very serious impacts on the integrity of the vessels and the health of the circulatory system as well. And also another big facet could be tissue engineering and regenerative medicine, where there's a lot of work as well in terms of bioengineering technology. So just to think about these problems and what might be important aspects of blood flow to consider, I want to start with a very basic question, which is to develop a simplified expression for the velocity profile of blood flowing through a vessel. So as usual with engineering problems, we're going to draw a picture of what we're talking about. So I'm going to imagine the vessel in this context to be a cylinder whose axial direction is along the z direction. We have a radial direction in the cylinder as well, and then the theta direction is circumferential. And again, as we typically do in engineering, we're going to make a number of assumptions to simplify the problem. That's going to help with our ability to solve it. And then later on, we'll talk about how we could potentially relax some of those assumptions as we add complexity to our system. So some of the assumptions we're going to make, or all of the assumptions, will be that the flow is fully developed. So what that means for pipe flow in particular is that when a fluid enters a pipe, it takes time for the presence of the boundary to be felt by all of the flow. So in particular, a little fluid element in the middle might not realize that you've entered a pipe. So it takes time for that information to reach that point. And so the amount of time that it takes is called the entrance length. But beyond that, everyone is aware that we're in a pipe now. This is how we're flowing. And so the flow can be considered fully developed. Related to that, we're assuming that the flow is at steady state. We're assuming that there's no circumferential flow. These sort of build off the prior assumptions. The flow is incompressible, which for water-based things is quite a good assumption, but as we know from earlier fluid classes, even compressible fluids can be modeled as incompressible as long as we're not too fast, so usually below a certain Mach number threshold. We're going to assume in this context that we have a constant axial pressure gradient, which I'll revisit in a second to explain why. we're going to assume no slip at the vessel walls. We're going to assume we have constant, a rigid constant diameter cylinder of radius r. And finally, we're going to ignore gravity. So we're in a magical world where everything's flowing horizontally along the z direction and we don't have to worry about gravity. So let's build some intuition, as I think that's a pretty good place to start for any engineering solution problem. And the best, or one of our key ways to do that is through drawing a free body diagram. So now I've stretched out the cylinder a little bit so we can focus on a small length L. And so what we have on one side would be a pressure, say on the left-hand face. The other forces that are acting on the system, or pressures or stresses, which we'll convert back to forces in a second, would be pressure on the opposite face, which will be P1 plus some change in pressure delta P over this length L. And then the other forces acting on the system, or stresses, would be viscous shear stresses along the wall that are resisting the flow. So as usual, we're going to sum all these forces. And so writing out the force balance, we get that these pressures are acting in opposite directions, and we have to multiply them by the cross-sectional area of the pipe, which is pi r squared. And then we're also having resistive force coming from the viscous shear stress that acts over a slightly different area, which is the area of the inside of this element of the pipe now. So it's not the cross-section, it's 2 pi r L. So we can rearrange this expression, as I mentioned, so P1 is the pressure driving the flow, and then delta P is the change over this small element L. We can rearrange this expression to get the following result, which allows us to write this pressure change in the following way. And this is interesting because we learn a couple of things. First of all, this pressure drop is negative, so the pressure is decreasing along the flow direction. And also, even for conservative or small values of the wall shear stress, when we're in a regime where the length of the pipe is much greater than the radius, which is likely true for blood vessels, which we said were, hugely long but quite small in radius, this pressure drop can be very large. And one last time, rearranging this in terms of a pressure drop per unit length, what's interesting is we see that this actually does not depend on your radial position or on your axial position either. So let's start to solve these equations. So to do so, we need to turn to Navier-Stokes equations, which you've seen in prior intro fluid mechanics classes. So the first equation that we're going to consider is the continuity equation, which is derived from the principle of mass conservation. Now for an incompressible flow, which means that density doesn't change in time or space, we can rewrite that to say that the divergence of the velocity vector is 0. And I'm sorry, I chose cylindrical coordinates. They are not my favorite coordinate system either, but rewriting this expression out fully, we get the following result for the divergence of the velocity vector. So some of our assumptions that we made previously were that the flow is fully developed and that there was no circumferential flow. So this tells us that there's no radial component to the velocity and no circumferential component. So that means in this equation, we can eliminate these two terms, which I'm showing you with my cursor. I hope that's visible. Oh, actually, I can do this, which is even better. Can eliminate the UR and the U theta. And so what we're left with then is that the derivative of the axial component of velocity along the axial direction is 0. And so that tells us that even though we don't know the exact form for the velocity vector yet, we know that it only has an axial component and that this axial component is only a function of radial position. So the next component of the Navier-Stokes equations would be the momentum equation, which in full form is written in the following way. But again, turning to the assumptions that we made, which is that we have steady state flow and a constant axial pressure gradient and that we can ignore gravity, we're only left with the following terms, where we see that the pressure gradient in the z direction is balanced by the divergence of the stress tensor. And here I'm just writing out for you explicitly what the stress tensor is, just a reminder of what I mean by these double underlines here. So essentially it has components from the radial, the circumferential, and the axial directions. So this looks pretty good, but this is in terms of a stress and not a velocity, and we're trying to find a velocity. But before that, one thing that sort of now becomes a bit clearer is what this pressure gradient means, and it's related back to what we found before when we were building some intuition from the free body diagram, which was this change in pressure over the length L. And this motivates a little bit for you why we can take it to be a constant in this scenario. But now what we need to do is we need to find a constitutive equation to relate this stress tensor back to the gradients of the velocity field if we want to solve for the expression for the velocity field. So the easiest relationship that we can come up with is what's known as a Newtonian constitutive equation. So this tells us that the stress is directly proportional to the strain rate tensor, which is related to the velocity gradient tensor, through a constant proportionality factor, which is known as the viscosity. So here, if I use the Greek letter mu, that means it's a constant viscosity. So for our flow field, which we found has the following form, we're left with the following expression for the divergence of the stress tensor. So this is a little quick, I would write out all these terms for you if I had a blackboard, but also you probably wouldn't want to see that because it's a mess in cylindrical coordinates, but just take my word for it that once you eliminate all the terms that are zero from the radial and the cylindrical components, this is what you're left with in terms of the divergence of the stress tensor. And then we can plug that back in to our momentum equation, and we see now how the pressure drop is balanced by this particular component of the stress tensor divergence. So now all we have to do is solve this. So we can integrate a first time, since we know that the axial pressure gradient is constant, and use our boundary condition of the fact that the velocity has to be finite at the center line, and then integrate a second time using our second boundary condition of no slip at the vessel wall. And we're left with the following expression for the axial velocity profile as a function of radial position. And just to sort of make this feel a little bit more tangible and related back to the engineering concepts that motivated it in the first place, I'm just going to re-express this quickly in terms of the average velocity along the pipe, which I remind you we calculate by integrating this over the cross-sectional area of the pipe. And now we see that the average velocity is related to the pressure drop, the radius of the vessel, and the viscosity of the liquid. And just pulling up some data from a paper on perforating arteries in the brain, which gave estimates that vessels in this region might be on the order of about 150 micron in radius. These experimentalists measured a blood flow of about 1 centimeter per second. And here I'm going to assume that blood has the viscosity of water, which we're going to revisit in a second. This gives me a pressure gradient of about 3,500 pascals per meter. And just plotting what this looks like, what this profile looks like using these numbers, we see that we've recovered what is known as Poiseuille flow, which is a parabolic profile where we have fastest velocity along the center line of the pipe that drops off to zero at the edges. Okay, so that's what we get when we treat blood as essentially water, and we look at it flowing under very simplified conditions in a cylindrical pipe. Now, let's think about whether the viscosity assumption that we made is a good one for blood by thinking a little bit about what blood is made of. So the background is fluid is plasma, which is an aqueous saline solution with proteins such as fibrinogen and albumin and globulins, and then there are also a lot of cells in the mix: red blood cells, white blood cells, and platelets. So this little schematic that I drew is actually not very informative of just how dense the number of cells is. So this is a little cartoon that I found of red blood cells in a vessel, and this can be quantified through what's known as the hematocrit, which is the volume of red blood cells to the total volume of blood. And in adult humans, this is about 30 to 50%. So this is telling you that red blood cells are really taking up a substantial portion of the total blood volume. So maybe we want to reassess how we think blood behaves when it flows. So to do that, I introduced rheology a little earlier. So I'm going to just walk you through a bit of a hypothetical rheological measurement of blood, just to see how we could quantify the viscosity a little bit better. So we're going to use what's known as a rheometer in a cone and plate setup. So we have a very shallow angled cone up here, and then we're going to put blood here. So maybe this should look red and not blue. And then what the instrument measures in terms of torque can be related back to the fluid stresses. And so what we're going to do in this experiment is we're just going to turn the plate at a constant shear rate gamma dot. And from the stress that we measure in the fluid, we can back out what the viscosity is at that shear rate. So as I said, let's turn on the rheometer. We're going to apply a certain shear rate. We're going to wait till everything equilibrates and measure the value of stress. And then from that, we're going to say, all right, from this relationship, we know that this was the viscosity of that shear rate. If we're dealing with something like water, which is Newtonian, then at the next value of shear rate, we would measure the same value of viscosity. So independent of how fast you turn this plate, the material is always going to have the same viscosity. Now, for something like typical biomaterials or things that are known as shear thinning, at the first shear rate value, we might measure a certain viscosity and a certain stress. But as we increase the shear rate, what we would see is that the viscosity starts to drop. And mechanistically, frequently what that's attributed to is the components of the fluid starting to align in the direction of flow and starting to deform and stretch out, and overall contribute less resistance to the flow. So here is what the viscosity profile of blood would look like as a function of shear rate, which I took from this paper published a few years ago. And what you can see is that it does shear thin, so the viscosity is lower as you increase the shear rate. And interestingly, this was a really nice paper from the 70s that started to explain why that looked into explaining why this might happen for blood. And so this experimentalist took 3 different conditions that he measured with the rheometer. He took normal red blood cells in heparinized plasma, which was supposed to represent what sort of physiological whole blood might look like, and that's this solid curve. He took normal red blood cells in a Ringer solution, which prevents aggregation between red blood cells. And then he took hardened red blood cells that can no longer deform in the same Ringer solution that prevents aggregation. And so by doing this experiment that I showed you before on all 3 solutions, what he shows is that this increase in viscosity at low shear rates is likely due to aggregation between red blood cells and the formation of these rouleau shapes. And then at high shear rates, the drop in viscosity relative to this hardened solution where there's no deformation is likely attributable to the red blood cells starting to deform and align with the flow. And interestingly, hardening of red blood cells has disease applications as well in conditions such as sickle cell anemia, where the red blood cells become hardened and not unable to deform, and that can cause a host of circulatory problems as well. And so what this tells us is that, sort of just quickly Googling what physiologically relevant shear rates might be, we're likely going to be in the range where shear thinning is important. And in particular, rheological plots are generally plotted on log-log scales. So even though it might not look like a huge change, this is sort of more than an order of magnitude in terms of a viscosity drop, which is substantial. So there are different ways that we can then, as rheologists, incorporate this non-Newtonian behavior. Some of these approaches include ground-up approaches where we try to model from a physics perspective all contributions of the material to the overall viscosity response. But another approach that we're going to use today, which is a little more simple, is to use what's known as a generalized Newtonian fluid model, or an empirical expression for the viscosity, which we develop by fitting to experimental data in order to come up with an empirical expression. So one of the simplest ones that exist is known as the power law model, which was proposed by Ostwald in 1929. And you can see here that it assumes that the viscosity depends on the shear rate through a front factor and an exponent. So I took this model and I fit it to the high shear rate data that I showed you before for blood. And this got out that the exponent was 0.8. So plugging that into this expression, 0.8 minus 1 is negative 0.2. So we see that, again, this predicts decreasing viscosity with increasing shear rates. So it might jump out at you. There are some limitations to this model. In particular, what would happen if you plugged in a value of 0 into this expression? You would get an infinite viscosity prediction. And if you plugged in a very high shear rate value, you would get a prediction of 0 viscosity. So again, as usual, at the expense of simplicity, we have to be careful with the applicability and the assumptions that we have to make, but this provides a nice starting point for starting to incorporate non-Newtonian behavior into our fluid flow model. We can increase the complexity, say in models like the Caro-Yasuda model, where Caro was actually from Montreal and I think still associated with UDM. So just a little Montreal context for you there. So now we've increased the number of parameters that we have by more than a factor of 2, but we can achieve some more interesting dynamical rheological behavior, like the appearance of a high shear rate and a low shear rate plateau viscosity, but at the expense of parsimony. So as usual, that's the trade-off. So this is where I left off before in the Newtonian solution, saying that we needed a constitutive equation. So let's do that now using our empirical relationship that we found instead of the Newtonian constitutive equation, where I'm just noting here that the stress tensor is symmetric, since that makes our solution a little simpler. So now for a power law fluid, we can rewrite out what the relationship between the stress tensor and the strain rate tensor is, and our same flow field. Now we can go on to explicitly write this in terms of the power law model that we're using, where we just have to be a little bit careful because the shear rate here is negative. Sorry, the gradient of the axial viscosity is negative, since we found that it was fastest at the center line and slowest at the pipe wall. So as we increase in R, the velocity is decreasing. So we just have to be a little bit careful when we think about that, because if we plug in this expression into viscosity, we don't want a negative or a complex viscosity value. But in any case, making sure we do that carefully by taking absolute values, we get back to similar expressions to what we had before. And our momentum equation reduces to the following form, where here, just in the interest of making the calculus a bit less messy, I'm leaving it in terms of the stress component of the stress tensor for now, and I'll convert back to viscosity, to velocity after the first integration. So we integrate a first time, assuming once again that we have our constant axial pressure gradient, and we eliminate our first constant of integration, since we know that the flow velocity has to be finite at the center line. And then again, using the caveats that I mentioned before of making sure we don't get imaginary numbers here, we integrate a second time and we get the following expression, which we can again solve for C2 using our boundary condition of no slip at the vessel wall. And this gives us our final expression for the axial velocity as a function of radial position. And I'm just going to pull up what the Newtonian expression looked like for comparison. We see that there are similarities in terms of depending on the pressure gradient and the radius of the cylinder. But now in this power law case, the dependence on the viscosity is a bit more subtle, since it's separated into both this N and this M term. And plotting what this expression looks like for the power law model for the parameters that we found previously, we get the following velocity profile. So this might be a little bit anticlimactic, since it kind of looks reminiscent to what we saw before, but when you line them up side by side, you can see that there are some notable differences. In particular, if I draw your attention to the scale of the y-axis, we see that now incorporating viscosity or power law model for viscosity into the material, we see that the velocity decreases. And you can think about that by saying, well, if I had a pipe and I use the same pressure to drive both honey and water through the pipe, the honey, which has a higher viscosity, would be flowing more slowly because there's a lot more resistance from shear stresses of the wall. And not only is the magnitude of the velocity slowing down, but when we think about this in a non-dimensional sense, where now what this plot is doing is it's decreasing the exponent n as you move in this direction, you see that the flow starts to move from a parabolic profile to one that's flattening out increasingly and becoming close to almost a plug at the smallest values of n, where you've got quite a constant value of velocity over a wide range of the pipe and then a sharper drop off to 0 at the edges. So all this to say that changing the viscosity expression of the material can have quite a big effect on the expected velocity profile. So I'll wrap up there. I'm just going to leave off and say that we can add in additional biological complexities if we want. So, and we could think about what adding deformability to the vessel walls might do, which is more sort of in the solid mechanics realm. We could think about adding curvature and branching to the vessels, or non-uniformity in their diameter, which would have a whole host of other interesting applications to the velocity profile. So probably you couldn't just solve all of this on a piece of paper, you'd probably have to turn to some CFD or numerical solution analysis, but just to say that this sort of would motivate that kind of thought. And then of course we could think about a more realistic pressure, which could be a pulsatile flow as occurs in the body through the heart driving blood flow. So with that, I'm just going to leave up some summary points and some takeaways and further reading options, and I'd be happy to take any questions.
Thank you, Caroline. So, questions from the students or pseudo students? And the students behave as usual, they do not put questions. If I may ask, just not a student question, did you use a textbook here from, or was that presentation coming from a particular textbook?
No, I mean, I verified the solutions for power law fluids from different textbooks and stuff, but this was just, I mean, I wanted to talk about rheology, and so I wanted to find a way to incorporate a problem that had non-Newtonian and non-Newtonian behavior. But
we have two questions. Look into the chat.
Oh yeah, here we go.
The
Why did I not consider gravity? Well, the short answer is I wanted easier integration. But yeah, that's a good point. So I don't think it would be a huge aspect to resolve that. Let me just try to go back to, oh wait, 1 second. So I excluded gravity because I was thinking about a scenario where I had a horizontal pipe, and in that sense, it's a safe assumption to ignore it. You could do non-dimensional analysis to think about that as well. But if I were thinking, of course, about a vertical, if I turned this pipe and I rotated it vertically so that now pressure was driving it upwards, then in my free body diagram, you're right that I would now also have an additional force coming down, which would be from gravity. So I would have to think about it in that sense. And you're 100% right that in the body, there are vessels that are pointing upwards, so gravity is likely playing a role. But another important tool for fluid mechanics analysis would be non-dimensional analysis, where you think about the relative contribution in terms of the magnitude of different terms. So in certain cases, it could even be justified to exclude gravity, even though we know it's a force that's always turned on. So that would be something that would be problem specific to think about it. But that's why I chose to ignore it in this scenario, because I'm thinking about this horizontal pipe flow. And then there was a second question too. Are Oswald and Karo-Yasuda both empirical? Are there physics-based models? So yes, these are both empirical models developed by essentially, I mean, they had physical intuition, of course. So for instance, Karo-Yasuda implementing this low shear rate and high shear rate viscosity is based on physical intuition of what's happening with the materials. There's definitely ways that you can try to recover these models by building up from the bottom the polymer physics of the different components of different biomaterials. And actually this is something that my group at MIT was trying to think about a lot, was because these power law models actually come up all over the place when we think about biogels and biofluids in terms of both G prime, G double prime, and viscosity. And so thinking about what sort of structure could give rise to macroscopic power law response, and whether that's sort of some sort of fractal structure, self-similar structure. So this isn't to say that you can't build things that achieve similar results from a ground up approach, but in terms of both of these models, yeah, they're empirically derived, or at least they were initially.
There is another question from Robert.
Yeah, I would say that it does indicate some problems, and in particular, you have to think about being careful with the range of shear rates that you consider, given the range of where your model is actually able to fit the data that you have. So if you wanted to really think about characterizing a flow at infinite shear rate, probably not the best to use an empirical viscosity model that is not valid there. But yeah, I mean, it's a subtle aspect that you need to think about carefully in order to achieve the right physics that you're looking for, but it still provides a real applicable answer at the end of the day.
Okay, so if there is no other question.
I have a comment.
Oh, sure, I mean, yeah.
No, I'm teaching the 340, and at the beginning it was involving the 314. This is a Bayern, actually. It's transport phenomena, and
Yeah.
I find, I mean, your delivery excellent in a sense that it was making the combination, actually, of the biological application with regard to what you can find in any book with this specific equation. So this is a positive comment. That's what I wanted to make from someone who suffered putting together a lot of slides to teach this course.
Okay.
That's it.
Glad to hear that.
Yeah, same here. So I had a 340 before. Okay, so with that, thank you very much, Caroline, and I see that you have a full day. So the two of us will meet towards the end of the day.
Sounds good.
Thank you all. Take care.
Bye.
Thank you all so much, Caroline. So I will now make you the full host.
Okay.
You can proceed with the rest of your meetings. Thank you all so much for joining. If you wish to share, if you know anyone who couldn't attend today's seminar, it has been recorded and I will upload it to our YouTube channel. Have a great day, everyone.